So I'm a PhD student in the Manchester Institute of Biotechnology, working in the Daniela Delneri's group. And yeah, I'm going to tell you a story about yeast, about hybrids, and about beer. So, excuse me. So first of all, I would like to actually define what's actually beer. So beer is basically, cereal juice boiled with hops. The cereal normally used is barley, and you boil barley with hops, and you obtain this product known as wort. Wort is basically what is fermented and transformed into beer. Fermentation happens thanks to the action of yeast. Normally, you might know Saccharomyces cerevisiae, which is the bakery yeast, is very well known and characterized. And what happens is you put your wort inside a fermenter, and fermentation happens. So basically, fermentation is the transformation of sugars into ethanol and CO2 by the action of yeast. So you start with wort, and you end up with beer. So as I just mentioned, Saccharomyces cerevisiae is one of many yeasts of the Saccharomyces genus, but you have others. So Saccharomyces cerevisiae is involved in the production of wine, beer, bread, and then you have some other Saccharomyces yeasts, like for example, Saccharomyces pastorianus, which is a hybrid in between Saccharomyces cerevisiae and Saccharomyces eubayanus. The main difference in between Saccharomyces pastorianus and Saccharomyces cerevisiae is that Saccharomyces pastorianus is involved in lager fermentation. It likes it cold, and whereas Saccharomyces cerevisiae is involved in ale fermentations. Another difference is when you have an ale fermentation, the yeast stays on the top of the fermenter, and on a lager fermentation, the yeast stays on the bottom. And then you have, for example, other Saccharomyces species like Saccharomyces uvarum, which is involved in wine fermentations. But as I showed you that the Saccharomyces pastorianus is a hybrid, but you have a huge variety of hybrids inside of the Saccharomyces genus. So you have basically everything hybridizing with everything and producing all kinds of different products, from wine, cider, beer, etc. You can have even hybrids formed by 3 different Saccharomyces species. And now I'm going to talk about a bit more in depth about one of the other Saccharomyces species, Saccharomyces uri. So Saccharomyces uri is a yeast species that was discovered by our lab in 2017. And I'm going to tell you how we discovered. So we have a field course that we organize every year on the French Alps, and where the students, are having fun, hiking, doing some climbing. The staff does actually research and work. So this is one of our former staff members, Samin and Asep, that was directly involved in the isolation of Saccharomyces uri. So just as an example, here I have a bunch of different isolates that we took from soil, from bark, etc., from bark. And what we did was we sequenced a genetic marker called the ITS region, which is widely used for identifying fungi in general. And we compared this sequence to databases and see how similar or different these sequences were. And we discovered that for a couple of them, the identity was actually pretty low. You might think that 98% is very high, but in terms of species, 98 means that might actually not be, in this case, Saccharomyces micati or Saccharomyces paradoxus. It might be a different thing. It might be a new species. But the thing is, how could you describe what species here is? So if we go to the very basic definition of species, you can think of it as, when you have 2 organisms that can reproduce and mate and generate, fertile offspring. Those organisms are considered to be of the same species. So if you have, for example, if we go to the hybrid sterility example, if you have a horse and a donkey, which are different species, and you mate them, you end up with a mule, which is sterile. So their offspring is sterile. So these 2 things are different species. So we follow this same approach to test if the new isolate was indeed a new species. So we took our potential Saccharomyces uri, the new isolate, and we crossed it with Saccharomyces cerevisiae, Saccharomyces micati, and Saccharomyces paradoxus. Three very related, very close, let's say, brothers in the family tree. And indeed, none of the hybrids that we created were able to generate fertile offspring. So this is just some of the data on the spore viability of these hybrids, and was very low. So this means that Saccharomyces uri is not able to mate with anything that is similar to it. So we consider it is a new species. Then we did some follow-up work, and we sequenced the whole genome of Saccharomyces uri, and we confirmed that it was indeed a new species. So yeah, we have a new species. But now we have assessed that we have a new species, but what can we do with this new species? So for knowing what can we do with it, we need to first to characterize it, right? To know where does it like to grow, what kind of temperatures it tolerates. So first, we characterized the optimal temperature that it likes. So this is just a plot showing you the optimal temperatures of all the different Saccharomyces species. On top, you have Saccharomyces cerevisiae, which likes it very hot, above 30 degrees. And then Saccharomyces cudatzevi likes it quite cold, below 20 degrees. And Saccharomyces uri sits near of Saccharomyces pastorianus, which is the yeast I just mentioned before that is involved in lager fermentations and likes it very cold. So Saccharomyces uri likes it cold. And then, not less important, is to assess the, what kind of food does this yeast eat? What does it like? So we assess many different foods, many different carbon sources to see what was being metabolized by uri and whatnot. And something quite remarkable is that Saccharomyces uri was very able to ferment maltose, maltotriose, and glucose. And interestingly, these three sugars are the main components of wort. Wort is this sugary substance that is used for making beer that I just mentioned before. So we thought, wait a second, we might be able to do beer with this yeast. So yeah, one potential application is brewing. However, we have to have in mind that Saccharomyces uri was isolated in the forest, in the wild. So it's not going to be adapted to grow in wort. It's been living in the forest for probably millennia, so it's not going to be growing very well. So since we wanted to make beer with it, we thought, why not hybridize it with a Saccharomyces cerevisiae strain that is used for brewing, a commercial beer strain. So we made a hybrid. And you might think, why should you make a hybrid? So the main thing about hybrid is that in a hybrid you have the genomes from both parents. And we have a genome, when you have two genomes, there is a lot of genetic variation inside the same organism. And this huge genetic variation can be very beneficial because the organism might have advantageous traits for one of the parents over the other. This is very, very commonly used in agriculture. Most part of the crops are hybrids because of the yield. The yield of hybrids is way bigger than the yields of the parents. And also, something to consider is that since the genetic variation, the genetic material that is inside of these yeasts is bigger, let's say the deck of the cards that they play is bigger, evolution has more room to act upon. So with bigger genomes, there is more chance for evolution to do something and be beneficial. So for example, something quite remarkable is that hybrids can adapt very well to sudden changes in environments, way better than the parents. This is a classic example of the pinches from Darwin. And focusing a bit on yeast, as I just mentioned, in a hybrid you have the genomes from two parents, and these genomes interacting between themselves. But you also have the proteomes of both parents, which proteomes, I mean the sum of proteins that is present inside the cell. Proteins are encoded by the DNA and expressed and present in the cell. And both the genomes and the proteomes can interact in between themselves and generate a wide range of genetic interactions and phenotypes. So how can we take advantage of this huge rainbow of phenotypes that we can obtain when making hybrids? So going back to the story of uri, we took advantage of this by making 3 hybrids. And one of them, this is basically just for you to understand, it means the amount of sugar present in the fermentation. And you see, and this is the time course. So the sugars go down together with the time. So the less gravity, the less sugars present in the fermenter. So this one, the blue one, is Saccharomyces control. So Saccharomyces, what is the Saccharomyces parents, the brewing strain. This is Uri, the red one, and all the other three are three hybrids. And for one of them, H3, we see that it completed the fermentation pretty much the same as the Saccharomyces cerevisiae brewing strain. So yeah, very well behaved. It produced a nicely attenuated beer. And then, but we don't care only about having a beer with alcohol. We want this beer to taste good. So we assessed the aroma profile. I won't go into detail, but this is basically as an overview of all the different compounds that play a role in the aroma and the flavor that you have in the beer. And the amount that was produced by Uri and about the one that was produced by the other parents. And we discovered that when you put Uri in the equation, you have a beer with a distinctive clove-like spicy flavor that might be desirable for some people and can be commercialized. And we did commercialize it in collaboration with Cloudwater. We released, Cloudwater released 1 month ago the new beer made with this hybrid H4 called Tales from the Future, which is this beer here. Yeah, it was quite exciting to see, the application of yeast isolated from the forest to biotechnology and see it in a bottle and sell as beer. Okay, so going back a bit to the hybrid story, there is some other work that we've been doing about, as you just mentioned, when you have a hybrid, the progeny, the progeny is sterile. So that's a bit of a pain, because when you have a hybrid, the genetic variation is huge. But it's kind of a shame that you kind of close the door for more recombination to happen, because these guys are sterile, so they cannot produce any offspring. So what we did was restoring the fertility on hybrids. And well, the main cause of sterility in hybrids is because the genetic material from both parents is not compatible, so they cannot recombine and produce offspring. But if you take two hybrids from two different species, and you combine them in a tetraploid, making sure that you have two pairs of chromosomes that are compatible between themselves, you can produce a hybrid, a tetraploid hybrid that is fertile. And what can you do with it? You can generate an even broader range of phenotypes very, very easily. With phenotypes, I mean just how well or how bad they grow in certain conditions. So that's what we did. We generated fertile progeny from tetraploid hybrids. And this is basically, so each dot is one of the sons. And these are growing in different conditions, in different temperature conditions. And the lower means the worse they grew, and the higher the best they grew. This is the colony size on solid media. So basically, we want the ones that grow very well in whatever conditions we're interested in. So this process of making hybrids making hybrids non-sterile, fertile, allows us to generate a lot of potential candidates which were very fit in a broad different range of conditions. So I'll be talking quite a lot about the Saccharomyces species, Saccharomyces genus, Saccharomyces cerevisiae, and how broad and how diverse only this genus can be. But can you imagine, because we're focusing only in one very small part of fungal diversity, but there is a huge amount of non-Saccharomyces yeasts, non-conventional yeast, still present in nature for us to go and grab if we want. So, because, for example, coffee, chocolate, lambic wine, and cider, all of these products are normally produced thanks to the action of spontaneously present fermentation. So this is all spontaneous fermentation. So these spontaneous fermentations can be a great source of microbes, of non-conventional yeasts for us to go and grab. So I'm going to tell you a bit about a project that we've been developing with a cider farm in the southwest called Thor Cider Farm. So these lovely people allowed me to go there the last pressing season, last year. And I went there and tried to, try to assess the microflora and all the wild yeasts that were present and involved in the cider fermentation. This is a wild fermentation, traditional scrumpy kind of cider. So I isolated yeast from the apples, the machinery, the fermenters, the tools, the ceiling, everywhere, to see if the yeast presence in their surroundings were actually playing a role in the cider fermentation. And they indeed were. This your left pie chart is just an overview of the huge diversity that we isolated from across all the cider farm. And this is the definitely more small diversity that was involved in the actual cider making process. So, again, as with Yuri, we have dozens of isolates, and we don't know how well and how bad they behave in different conditions. So we assessed their fitness, their growth, in the different conditions. In this case, in the cider making conditions, in conditions that might be relevant for the cider makers. So we assessed how well they grew in the different sugars, sucrose, fructose, glucose, and also ethanol concentrations, increasing concentrations of ethanol. And across more than 100 isolates that we studied, there was a bunch of them that in general were behaving very well across many different conditions. So all of these candidates might be very nice for trying to make maybe more controlled cider fermentations or fermentations of other uses. But again, as with Yuri, we needed to know what was the aroma, the flavor compounds that these guys were producing. So we did 23 different single strain fermentations in the lab and measured the aroma compounds. And well, this is a bit overwhelming, but I will just focus on these two. So for example, for this compound that produces a fermented banana kind of flavor, we saw that Hanseonospora species, Hanseonospora ovarum, were generating a lot of this compound. So if you're interested in a beverage, in a cider with like very fruity aroma kind of flavor, banana kind of flavors, you might want to use this species specifically. And then, for example, another compound, ethyl acetate, which has also a ethereal, fruity kind of flavor. Same story for Hanseonospora ovarum, produce quite a lot of it. a lot of it. And then also Pichia cluveriae, which is one of my favorites, also produce quite a lot. So same thing. These might be very good candidates for making controlled fermentations and trying to generate a product more reproducible, because when you do spontaneous fermentations, the product can vary completely from batch to batch, and cider makers sometimes want to be able to replicate the same product over and over. That's what is done in industrial cider making. They didn't use spontaneously fermented apple juice. They inoculate their apple juice with something that they know. So we can use this in single fermentations, or we can even try to do co-fermentations and mix different species, taking into account the aroma that they produce, to kind of generate a tailored end product depending on what kind of aromas we want in certain types of cider. So yeah, that's basically it. So take-home messages. As I just mentioned, there is a huge fungal diversity still there for us to exploit. Hybrids are a great way of increasing genetic diversity and increasing, generating potential candidates for a lot of different biotechnological purposes. And what we did of breaking a speciation barrier was a great way to even make bigger this diversity and be able to generate candidates even with more fit behaviors. And very quickly, I would like to thank all my group, the Delneri group, especially Federico Visnioni, which was involved in the hybridization, in the restoring fertility project, as well as Aminonacep. Constantina and Federico were involved in the Saccharomyces Yuri beer production in conjunction with Cloudwater, Mark and Paul. And I've been working in the last part that I just told you, the cider story, which was thanks to the collaboration of Thor Cider Farm and Singer Instruments, and with the help of Oliver Serban. Thanks a lot.
Fantastic presentation. You crammed in so much there, Jose. Can I just ask you, Before I open it up, what is the next stage? What are you hoping to do next?
So that's a very good question. So we have this bunch of isolates that come from the Sire farm, for example, and some of them produce a very nice aromas, but don't tolerate very well certain conditions like ethanol. So we are trying to do something which is non-GMO, which is random mutagenesis, to increase the tolerance of this species to ethanol, to then have yeasts with very high tolerance to ethanol, which produce a lot of good aromas as well. That's one of the things we're currently working on.
Okay. Has anyone got any questions for Jose? Well, the other thing I just wanted to pick up on was you said earlier on about how long brewers have been trying to find alternative yeasts like the one you discovered in order to continue finding new variations. So what exactly, is it a technological breakthrough that has allowed you to discover this now? Is there a bit of luck involved? Why have we been able to find this new variety, this new strain that is suitable now rather than, say, 50 years ago, do you think?
I would say it's more, obviously, technology has increased a lot in the last decades, and that helps in all kinds of biological research. But I think it's more in terms of customer preference. So in the last decade or so, craft brewing has been flourishing a lot, and customers of craft beer, they are bored of lagers and these ales. They want something different, something more interesting. So I think it's mainly generated because of the interest in the consumers.
So that commercial imperative too, then, that is driving desire for more research, effectively.
Yeah, I think so. Yeah, it's the main force.
And is there a way of quantifying at all? Then you said about how much effectively there is still left to discover. Is there a way of articulating what percentage even of microbiology we understand at this stage? Do you think?
I think there is some estimates. I wouldn't know exactly at the top of my head, but we're talking about we know like 2% of the diversity, the fungal diversity. So it is very hard to assess because a lot of these isolates don't grow in lab conditions. So you only rely on massive, taking samples and sequencing the whole thing, but that's very expensive and very time consuming. So yeah, it's very hard to define, but it's definitely huge diversity still for discovering.
So that is one of the main barriers at this stage then, is even if you are aware perhaps that a species exists, trying to investigate the extent to which it can be useful is a really time-consuming process.
It is very time-consuming. We, in Singer Instruments, the company that I work for, we develop robots and, high-throughput screening platforms and robotics accelerate this work, but these robots are very expensive and, funding is quite limited. So, yeah, we rely quite a lot on automation.
And then, so for people here today then, and for people watching on YouTube and other platforms later, sort of what is the sectors, if students are watching this, what should they be studying if they want to get into a field such as this? It's probably a combination of different disciplines, right?
Yeah, so you can end up here by studying biochemistry, microbiology, biology. I studied myself environmental sciences, and then I focused on the microbiology side. But yeah, we are very open to all kinds of researchers from a broad range of disciplines.
And do you intend to devote the rest of your career to alcohol?
Absolutely, yes.
Brilliant. Well, thank you very much for your time today. That was fascinating.